Now I have the opportunity to introduce you the last speaker of the session. It's Remi Villenave. And he got his PhD in Belfast at the Queen University. Then he moved to the States, to the Harvard University, and then to the Emulate's founding scientific team to further develop and apply the organ-on-a-chip technology. And indeed, the title of the presentation is Organ-on-a-Chip, a living platform for generating human-relevant data.
Thank you very much. I wanted first to thank the organizer for inviting me and giving me the opportunity to present our recent development on organs on chip. So first, let me introduce you to organ on chip. So these are small microfluidic devices. As you can see here, they are made of transparent polymer. They are called chips because they are microfabricated using fabrication methods from the microchip computer industry. And as you can see, you have hollow channel inside that contain the cells. It can be human cells or animal cells. And these cells will reproduce or simulate the physiology and function of human organs in vitro. So how do we do that? Because these chips are microfluidic devices, we can control the microenvironment of the cells and therefore reconstitute the tissue-tissue interface and human tissue structure and function. And we apply this to test drugs or agrochemical compounds, toxicity or efficacy in a human-relevant model. I'm going to go through a few examples of the chip we developed recently. This one is the lung on a chip. Or more alveoli on a chip. It has been published in Science in 2010. It was done by a postdoc from the lab I'm from at the Wyss Institute in Harvard. And the idea here is to reconstitute inside the chip the tissue-tissue interface of the human alveoli or air sac. You can see here the extremity of the respiratory tract, the alveoli, where the gas exchange between the air and the blood occurs. And you can look at this TM image here, and you can see the alveolar epithelial cells here are separated from the capillary cells, the blood vessel cells, by a small interstitium. Another component as well of the alveoli is that they are stretching cyclically due to the breathing. And therefore, we try to reproduce these three different characteristics of the alveoli inside the chip model. So the tissue-tissue interface between epithelial cells and endothelial cells, the dynamic flow of the blood under the capillary cells, and the cyclic breathing movement. So just this is to show you a little animation to show very simply how the chip look like. So you cut through the chips, and you can see that inside you have three channels. The middle channel is separated in the middle by a flexible porous membrane. And onto this membrane, we're going to see the cells, so the lung cells in this case, and underneath the capillary cells. And by applying vacuum on both sides of the channel, you can see that you can stretch the cells and apply a mechanical stress onto them the same way they will experience into the alveoli. You can also, as I mentioned earlier, put some air on the top and flow growth medium to mimic blood flowing in this case. So how does it look like under a microscope? So it's you have here the epithelium stretching and the endothelium stretching. And in this 3D reconstruction, you can see that you can recapitulate. You can form two very tight monolayers. So in green, the epithelial cells, and in red, the endothelial cells separated by this porous membrane. So we first wanted to apply this chip to mimic lung inflammation. So what happened during lung inflammation? Quickly, you have a pathogen that enters the airway. Here, enter the alveoli. The cells will alert the other cells and the immune system by secreting pro-inflammatory cytokines that will in turn activate endothelial cells on the other side of this interstitial tissue. And the leukocytes from the bloodstream will adhere to these endothelial cells. and start to migrate through the interstitial tissue to the alveoli and to deal with the pathogen infection. And so to orientate you here, this is a view in classic fluorescent microscope. When you look at the cells here in a non-infected chip, and you have this white blood cells, in this case neutrophils that have been stained to visualize, that are not attaching to the endothelium. However, when you inflame the cells, the chip, the neutrophil will then attach very quickly to the endothelium and be recruited to the endothelium. Once they are recruited, they start to crawl at the surface of this endothelium until they find a pore in the membrane, in this case here in the hexagonal shape. And then they go through the pore, and what happens is you have here the bacteria in green and the neutrophil in red. They will start to engulf and kill the bacteria in the apical side of the chip. Some partners, some collaborators we have were interested in specifically mimicking and testing drugs for COPD and asthma and some infectious disease. And for this disease specifically, the airway epithelium is central for the lung pathology. So we started when I arrived, it was 3 years ago already at the Wyss Institute. We started to develop this airway on a chip that would recapitulate what you have in your airway epithelium. In this case, pseudostratified columnar epithelium with ciliated cells and goblet cells that will produce the mucus. So this is what we wanted to achieve in our chip. So we used the same technique, a very similar design. Here you have the chip. And we slightly modified the design to make the top channel a little bit taller to accommodate the thick airway epithelium. And then we seeded human primary airway epithelial cells on this, on the porous membrane, and underneath, same human primary endothelial cells. And when we did that and cultured the cells at the air-liquid interface for 3 weeks, we recapitulated a cilia formation, the same cilia as present on the airway epithelium of your lungs. These cilia in the chip were beating the same way they do as in vivo. And when we introduce fluorescent beads inside the chips, I'm not sure it's very clear, you can see that the combination of the cilia beating and the mucus secretion start this mucociliary transport that is visualized here with these fluorescent beads. So this is just a table to recapitulate few parameters and compare between human and chip, what you have in human and what we found in chip. And you can see that it's very similar for many parameters, the percentage of cells, the cilia beating frequency, and the mucociliary velocity. So using this chip, we recreated some key features of the human airway physiology and function. And we then wanted to apply that to the disease I mentioned earlier, COPD and asthma. I'm going to show you, before I show you some COPD results, I wanted to really highlight the tissue-tissue interaction and why this tissue-tissue interaction, why having endothelial cells underneath epithelial cells is important in my opinion. So just to orientate you here, this is the 3D reconstruction of the airway on a chip, and you have the epithelial cells in green on the top, and underneath the membrane, the endothelium. And when you stimulate the epithelial cells or the endothelial cells alone, you get a normal pro-inflammatory response. We stimulate with poly-IC in this case that mimics a virus infection. And when you have endothelial cells or epithelial cells alone, you get secretion of pro-inflammatory cytokines, IP10, RANTES, IL-6. But when you have both at the same time inside the chip, you get a synergistic effect, a much higher secretion of these three cytokines. So there is really a crosstalk between the epithelium and the endothelium, and this crosstalk plays a critical role during lung inflammation. So to study COPD exacerbation, we use the same chip, same similar design, but in this case we use primary cells from COPD patient. We stimulate them with poly-IC to stimulate a virus infection, so an exacerbation. And we also, in this case, flew neutrophils, stained neutrophils in the bottom compartment on the chip. And what happens when you flow neutrophils in unstimulated chips? You have a basal level of attachment, but once you stimulate with poly-IC, you get a much higher increase in neutrophil recruitment to the endothelium. And of course, our pharma partner were interested to see if their drug could pharmacologically modulate this neutrophil recruitment. And we used as a control budesonide that is given routinely in clinic to a COPD patient. And we didn't see any decrease of the attachment of neutrophil. However, when we used their compound, we saw a dramatic decrease in the neutrophil attachment. Something completely different, we also wanted to use the chip as an infectious disease platform. I'm a virologist by training, so I was very interested to see if this chip could support virus infection. And you have here, because these chips are transparent, you can image them in real time under the microscope. And this is an infection with a Sendai virus GFP tag. Then you can follow the infection during 3-4 days, and you can really follow the virus infection and how it start to create syncytia and start to destroy the epithelium after 50-60 hours. Since it's a food-related conference, I also wanted to talk to you a little bit about the gut on a chip. So the gut on a chip is also the same model with the gut epithelium here. So you probably know much better than me the structure and morphology of the human intestine, but what we use here as a model is the gut epithelium. We use CACO2 cells. It was a few years ago, and we didn't have access to human primary intestinal cells yet. So we use CACO2 cells. We grew them onto the porous membrane and underneath with the endothelium. So same system. And we stretch them as well during 6 days. And what happens when you stretch them during 6 days? You start to have the formation of this pseudo-villi structure inside the chip. You have here a 3D reconstruction of the chip. And here is the SCM image. Maybe it's much more obvious here, where you have this structure, these formations here, and this canyon valley in between. And we use these chips to grow and to co-culture microcolonies of gut microbes. And it's a movie you don't see very well, but the bacteria are moving in between the villi and stay there for 6 days to 2 weeks. And because there is a flow underneath the cells and also on the apical side, the bacteria are not really overgrowing. They are staying there, and probably the excess. What happened is probably the excess growth of bacteria is washed away by the flow. We also have a collaboration with the FDA to test the gut on a chip as an enterovirus infection model. And as a model virus, we use Coxsackie virus B1, which is an enterovirus, to see if we could infect the chip and create a cytopathic effect. And indeed, after 24 hours, you can see that the chips start to lose these villi-like structures, and after 48 hours, the whole epithelium is obliterated. You can see it in this confocal picture that the virus is there and destroy the villi-like structure. What's interesting here is because this epithelium is polarized, we were able to see a polarized release of the virus into the apical luminal side. And even when the chips were infected basally, the virus was secreted apically. And this is also the case for the release of cytokines. As you can see here with IP10, there is a really polarized secretion of cytokines. So we have, I showed you 3 different chips, but there is also different other chips such as the heart chip, bone marrow, liver, blood-brain barrier, skin on a chip, thrombosis, muscle, and many disease models that we are developing at the moment at Emulate and the VIS Institute. And the application, I'm going to be quick here. So it's, just to give you an idea, it's quite a low throughput system at the moment. It's still in the pioneering state. And I would say, to be realistic, you can maybe culture 100 chips at the same time. One person can do 100 chips at the same time. It's not nowhere near the high throughput screening that you've seen before. It's made for high value key decision making data. As I said, you can test drug and toxicity in these chips. But there are also limitations and challenges. The fabrication is a challenge. We are at the moment discussing to outsource the fabrication of the chips to make much more like we have a collaboration with Sony, which makes DVDs, and they can fabricate 1 million of chip in 24 hours. But of course, we need to use them and at the same time. There is a lot of engineering problems also in such as bubbles, formation. The supply human cell is also an issue, but we are tackling this problem with the use of iPS cells. And I want to show you here to finish the human on a chip, a very pompous title for just linking different organ on chip together. Because of their microfluidic nature, they can be linked together, and the effluent will go from chip to chip, from the upstream chip to the downstream chip. And you can imagine that by treating the lung chip with a drug, you can see the effect on the liver or on the excretion with the kidney chip, and see the contractility effect on the heart chips. Same with the gut chip here. And this was part of the Vis Institute was part of this big DARPA initiative that was awarded $37 million to connect 10 different organ chips for 1 month. Another direction is personalized medicine, and we are collaborating with Cedars-Sinai in Los Angeles. They have a huge bank of stem cells, and they are really expert in iPS cells. Because, as you know, different population will react differently to drugs, and we are starting to use iPS cells to differentiate the chips and test drug in different population. Thank you very much.
Thank you, Remi. Impressive images, I should say. I saw 2 questions.
Yeah.
So how do you get to cells? So you have to get biopsies. So if you want individual screening.
Can you use the microphone?
Okay. So the question is, if you want to do individual screening, how do you get the cells?
So as I mentioned, there's iPS cells that we get from patient. The airway chip that I showed was with cells from commercial vendors, so they are readily available to buy from vendors, primary cells. You mentioned biopsies. That's also one of the direction we take. We also are going to use, I show you results with KCO2 cells in the gut chip, but we also use organoids, so cells from organoids that we seed inside the chips. So there is a
Homogeneous, do they react if you compare batch to batch?
That's the problem with every primary cells that you get. Some patient will grow very well and differentiate very well. Some patient won't. You have the same issue with if you culture the cells in any other system, in Transwell or things like that. But it's quite good, I would say.
Hi, Harvey Kluhl, the Hamner. It really is impressive how you can reproduce normal biology in at the chip level of scale. As I recall, that DARPA funding had a goal of also being able to produce in vivo relevant plasma concentration profiles of drugs. I'm wondering how that's coming along, because we have trouble at the plate level trying to get chemicals to get to the cells rather than be absorbed on all the tubing, and actually surface to volume problems increase as you go down in scale.
Yeah, no, that's a very, very good question. So I was not directly involved in the DARPA project, so I will have difficulties to answer. What I can tell you is that it's a very challenging project. We barely start doing some real toxicology and linking organs and in a relevant manner, not just linking organs and look at viability of the cells, but really putting drugs and seeing how they cross the gut epithelium, for example, and impact the liver viability or growth. So it's really in its infancy, I would say. I don't have a really good answer for this question.
Okay, there is another question here. It will be the last one, then we will start with the panel discussion. There.
If it's working, if I understood the message, I mean, there are good systems to study local effects of. In your presentation, the attention was mainly posed on pathogen. I wonder if you have some experience on chemicals, and then if you can study not only toxicity, but for instance, the cytotoxicity, or then when you apply potent mitogen, for instance, what happened? Because potent mitogen can preclude an adverse and inflammatory effect which can arrive to neoplasia, for instance. And can you use this model, for instance, to study genotoxicity? Because you are gut and lung, so they are very important.
Yeah, no, absolutely. So Emulate started earlier this year, so it's a very new company, and we are starting to get some grants to study drug toxicities, and our pharma partner are giving us drugs to test, and we also use chips with cells from dogs and cells from rats to compare also if the toxicity is different from chips with cells from human. So it's, I would say it's starting the toxicity studies. I don't have any results to show you yet, but it's definitely something that can be done, and that is being done at the moment, and will be done in the future. Yeah.
Okay, thank you again.